Now I wanted to put autotroph and heterotroph on here, but I can only have like 15 questions per chapter, you know, but I think autotroph is a very important term to know, but there was just so many things that I thought were important to, um, um, here it is, I closed it, okay. Okay, so now, I want you to know what aerobic versus anaerobic is. I want you to know the difference between aerobic and anaerobic. Okay, there are really no similarities, there are differences. Aerobic is any mechanism that requires oxygen. If it has a requirement, if it has a requirement for oxygen, and our cellular respiration, it requires oxygen. It's called aerobic. If it does not require oxygen, the environment does not require oxygen, it's called anaerobic. So aerobic requires oxygen. So our process of, and actually it's right here. If you look at this diagram, this is the electron transport system. And I'll talk about this in a minute. But this O2 right here makes this process, which is, glycolysis, how we process our food, the electron transport is right in here, right here. It's the latter part of the cellular respiration process, and it involves oxygen right here. This is what makes our eating process aerobic, because oxygen is required. And this oxygen in the electron transport system generates water. Okay, you still gotta drink water, but it generates water, okay? A water molecule is generated. So aerobic requires oxygen, and anaerobic does not, okay? And we have some aerobic processes. We actually have some anaerobic processes happening deep within our muscles, because our muscles are so compact. Well, not mine, but some athletes. Their muscles are so compact that,
Lactic acid?
Like who?
Lactic acid?
Exactly. You were listening, okay? But and the environment tends to be more anaerobic actually. But all my muscles are loose. Okay. So anyway, so that's the difference between aerobic and anaerobic. Now, I don't have any fermentation questions on here. I wanted to, it wasn't a test of your knowledge so much as I wanted to have on the exam things that I thought were critical for you understanding and taking to the next level, meaning another class, okay? And these are things that are bare bones information that you really, really, I think, need to know and understand. Okay, so for glycolysis, I remember I told you in class, you didn't necessarily have to know the whole process of glycolysis, but I did want you to understand what goes into that process and what comes out. The start of it, and it does, what is it? Did I close chapter 7? Yeah, I did. Okay, so this is the total process of glycolysis. For the total process of here it is right here. This by definition, this whole thing is cellular respiration. Anytime you see that word respiration, it means that respiratory molecules are involved. Your respiratory molecules are oxygen and carbon dioxide. So that means those two are involved, and they are. I showed you oxygen so far, and the carbon dioxide gets breathed out at various steps. At this step for the pyruvate, at the citric acid step, at that step as well, and you lose them all. They come out of your nose as carbon dioxide. So let's talk about this process, and I'll talk about them individually in a minute. But it happens in two phases. In the cytoplasm, the glycolysis, it's glyco, sugar, lysis, breakdown. And that's what digestion is. It's a breakdown process. You start with a 6-carbon glucose. You end up with 2 3-carbon molecules. And I want you to know what those are. They are pyruvate. So I want you to know glucose and pyruvate. You don't have to know all that other stuff that's in between. And you'll learn that when you get into biochem. Right now, you just know that you start with glucose and you end up with pyruvate. It's a 3, so you're starting that digestion process and you're stripping electrons from that glucose molecule. And you're giving it to NAD to make it NADH. And you're also generating ATP, adenosine triphosphate. Okay, all right, so, and that's here, and then after the glycolysis, you move into the mitochondria where all the heavy lifting starts. But at the pyruvate step, you actually lose 1 carbon. You lose 1 carbon in the form of carbon dioxide, and you end up with 2 2-carbon molecules. So I'm going from a 6-carbon molecule, glucose, to 2 pyruvates, to 2 acetyl groups. Okay? So I'm digesting away. I'm digesting away. And then that's that acetyl, it bonds. You don't have to know that necessarily. I want you to know that the 2-carbon acetyl group is then going to enter into the citric acid cycle. And when it goes into the citric acid cycle, it's going to bind to oxaloacetate. Okay, so it's a 2-carbon molecule that binds, okay, and we'll talk about that. But let me go through this real quick and then we'll go through the individuals. But I do want you to know what goes in. As it goes in as an acetyl group that binds to oxaloacetate. And then it becomes citrate. That's why they call it the citric acid cycle. In my day, we called it the Krebs cycle. Okay, so anyway, but I want you to know there's 2 ATPs right here. There's 2 ATPs right here. And then a lot of ATPs right here. And then by the end of this process, all you have a lot of energy. You have electrons and protons that you've stripped, and you've gotten rid of all the last 2 carbons that you have. The last 2 carbons in the form of this acetyl, you get it. We'll go over that. I'll show you that, and get rid of that. But then all you have is electrons now. You have electrons, and we don't talk much about the protons, but we're going to go into the electron transport mechanism, which is going to just transport those electrons to other molecules. And actually, I'm not going to go, I'll wait till I get on the diagram. I showed you that, the electron transport, they draw it like this with oxygen at the bottom, which then binds to, transfer electrons to a lot of different molecules, iron sulfate, cytochromes, and then some other things happen, and I'll show you that.
Are the diagrams going to be on the
That's a very good question. I generally don't put the diagrams on because my printer doesn't do color very well. Okay? So
Also, is the review in Blackboard?
I'm going to put it on Blackboard.
Okay.
I'm absolutely going to put it on Blackboard. HAB, what's your last name? H-A? Hoptom. I was gonna say happy. I was gonna remember I said you, because I'm notoriously screwing up people's names. I screw up everybody's name. I have a lab full of students, and they complain about it all the time. They just tell me, call them a nickname or something. There was one woman, Chinema, it took me half a semester to know how to pronounce her name. I got it right now, but it took me half a semester. It took me 6 months. I'm just bad with names. Okay, with enunciations. I just don't try. That's what it is. I mean, that's what it's about. I'm sorry.
Yeah.
I'm sorry, but that's what it's about. That's what it's being lazy. Okay, all right, so let's go back to, let's go through, and we're gonna go through these processes because I want you to know that for 1 molecule of glucose, which is this, all of this, one molecule of glucose you start with, you're gonna generate, 36 to 38 of these. And that's gonna depend upon the tissue, because, you talk about your brain, it's gonna be a little bit different, a lot different than, let's say, your hair or something. Not like your skeletal muscle or something. Well, some other organ, I can't think. but it's gonna be 36 to 38, okay? All right, so, and we'll talk about these individually. There are certain things I want you to know, about this process. I do want you to know electron transport. Here's oxidation reduction. This is I'm not gonna put any of the enzymes in there. Not gonna put any of those in there. But I do think that it's important to know that, and this is, and I'm just gonna explain this real quick, this is like representing the glucose molecule, just showing, you know, that things get stripped. You're literally stripping electrons when you go through a digestive process. And those electrons are temporarily given to NAD to make it NADH. So it's gonna be, you know, in the form of electrons and protons, actually. It's NAD to make it NADH for us. It's NADP to make it NADPH for plants. Okay. Okay, now let me, oh, here it is, right here. So here's here's the review. Okay, all right, let's see. I've already talked about 6 carbons to 2 3-carbon molecules. So you have 2 acetyl groups being generated. And I'll get to that figure in a minute. And I think I'm just gonna go, let me go back here. I'll come back here. This is that whole process. It's a lot of energy made from that. Let me go back. Let me go back here. Let me go back here. Okay, glycolysis, 2 ATPs, 2 NADHs. And then I'm gonna make sure I didn't miss anything. That's fermentation, actually. Decarboxylation, I don't need you to know that. Acetyl to, this is the figure I was looking for. And then I'll go back and make sure I didn't pass over anything in the review. But, so this is the citric acid cycle. So the acetyl is the two carbons right here. These are the acetyl groups. And in the citric acid cycle, it binds to oxaloacetate, which just gets recycled with the spin, but you know, the first molecule generated is a 6 carbon, 1, 2, 3, 4, 5, 6. And then it loses all the carbon dioxides right here. So I want you to know what goes in. What goes in is the acetyl group bonds to the oxaloacetate for the citric acid cycle. And by the time you finish this citric acid cycle, you have no more carbons. All you have is energy. And you have per acetyl group, and you have 2 of those. Per acetyl group, you have 1 ATP, 3 NADH, and 1 FADH. Okay, so that's per, so the arrow going this way means, you know, you're reducing these molecules. This becomes reduced, that becomes reduced, and the ATP, some figures just have ATP or GTP, same amount of energy, it doesn't really matter. And then NADH. So if you say, if I ask per glucose molecule, it's gonna be twice all of this. But per acetyl, which is the 2 carbon one, it's these figures right here. Okay, so let's go back to the review and see let's make sure we haven't missed anything. I have reduced and oxidized again. Know the where the citric acid cycle occurs and everything is now moved to the mitochondria. And then not so much you're not gonna have to know the structure of the acetyl group. It's just, I want you to know that it's a 2 carbon molecule. You know, it's generated from pyruvate. It's a 2 carbon molecule, and it's gonna bind to oxaloacetate. And per acetyl, yeah, and I say, I didn't want to give you the answer. I said, you know, determine how many ATPs in that per acetyl or per 2, or per glucose. Okay, the carriers that are carrying all this energy around, because there's energy in the electrons, is NAD and FAD. NADH and FADH, okay? And when you get to, I'm gonna make this, you can't really see it that well, when you go into, if you go from the citric acid cycle, so you don't have any carbons anymore, because you expelled all that out. And then all you're dealing with now is electrons and protons. And for the electron transport, you know, it's really, you know, in the membrane of the mitochondria, in the inner membrane, the couple of membranes and inner membrane space and all that. I just, you know, keep it simple, in the membrane. You know, there are these various proteins, flavoprotein, iron sulfate, ubiquitin, ubiquinone, and cytochromes. Now, I'm certainly gonna ask this question. And I mentioned that in class. I said, you know, when you compare the electron transport of our cells, and this is us processing our food, and electron transport for plants, have cytochromes in common. You actually have a whole bunch of proteins. It's not just these, it's a whole bunch. I don't know exactly how many. I think it's like, they say anywhere from 10 to 20. But for cytochromes, there's cytochromes in the plant electron transport, and there's cytochromes in our electron transport. That's something we have in common. There's ATPase, you know, which is functioning in chemoosmosis to generate ATP in plants. ATP synthase. Remember, whenever I say that, I say got the guy the Nobel Prize. That protein. Remember? And there's ATP synthase, which does exactly what the name says, which is to synthesize ATP. Synthesis means to make. So it's generating ATP using ADP as a substrate, a diphosphate making it into a triphosphate, okay? A di making it into a tri. And that is your money item. Okay, so anyway, for the electron transport mechanism, so what you have is, you have your electrons being temporarily transferred to these proteins. So I use in class I use cytochrome as an example. So you go from, you know, NADH to, so it gets oxidized. NADH gets oxidized to NAD. I had the reduced form before, now because there's a whole bunch of just passing electrons and passing energy around. And they call it the electron transport because that's what it's doing. It's transferring electrons from the NADH, FADH to, you know, I'll use cytochromes as an example, cytochrome H, cytochrome AH, cytochrome BH. Do you see the point? Just temporarily and then those electrons get trapped, you know, get transferred and get trapped within that inner membrane space. So it gets transferred here, but then it gets trapped in this region right above it. And that's what got the guy the Nobel Prize, defining how these electrons get trapped within this inner membrane space. Because there's another membrane right there, another membrane, and it gets trapped here once they get here. And I'll show you a diagram of that. Okay, so anyway, but the thing that makes this process aerobic, our process, our cellular respiration aerobic, is this oxygen right here and the carbon dioxide that's expelled right here. Okay? All right, and that oxygen bonds with hydrogen to make water, to generate water. Okay, so this makes it an aerobic process, and it's the electron transport. Is this a lot of material?
Always.
Is it beginning to get a little clearer?
I understand.
Okay.
I think.
I'm hoping that once you get the multiple guests, it won't be so bad. Okay? I'm depending on that. I'm depending on y'all to make me look good and have A's. Okay. All right. So anyway, so let's go with okay. So let's go to. My graduate students, for the first day, they are working by themselves. and I told them to get in touch with me if things go wrong. So far, everything's good.
Next thing you know, they're loud.
Okay, all right, so let's see, so, now oxidative phosphorylation, I didn't use that term a lot. Let's see, oxidative, that's the process by which, the ATP synthase generates all that ATP. The role of oxygen, I've already talked about that. The role of ATP synthase, I'm about to talk about that. But before I do that, let me just finish up with this, because I'm going to go to the slide that has the chemoosmosis on it. You get more energy from carbs or fat? Carbs. You know, you have more bonds, you got more energy. It's as simple as that. It's not complicated at all. The more bonds you have, the more bonds you can break, the more electrons and protons you can generate. And you get energy from all those electrons in the outermost shell. And sometimes you're breaking it down to the protons, and they have energy as well. Okay? So the more bonds you have, the more energy you have.
So carbs?
No. Fats.
Oh, I thought you just said that.
No. Fats have a lot. If I said carbs, no.
Okay.
No. But if
Sorry,
you
I guess I just heard wrong.
you absolutely get more energy from fats than you do from carbs. You do. It's got more bonds. They have more bonds.
Yeah, I thought so.
Yeah, they have more bonds. Okay? All right. If I said carbs, strike me.
You probably
But
did.
no.
I guess I
Okay.
just heard
All
wrong.
right. So now I wanted you to understand the process of cellular respiration. Understand it. You don't have to know it in detail. Understand to the point of what's on this. You should be absolutely A plus. Okay. All right. Now let me go to the chemoosmotic step. Okay. So this is that step. We've already talked about that. And I've already talked about 1 ATP, 3 NADHs, and 1 ATP FADH. If you want to know where all of this is going, I still have that slide on Blackboard where it has all the details with the carbons and the hydrogens. Very nice display, actually. Okay. And tell you all the bodies are buried, so to speak. Okay, now for the, we already talked about the electron transport. You're actually transporting electrons. That's why they call it electron transport. And it's going from NADH. It's being oxidized. And the FADH is being oxidized. And the membrane proteins are being reduced. Okay, and oxygen right there, very important. Oxygen, very important. And that's part of that electron transport. Now accumulation of protons and electrons in that innermost space, and this is kind of a diagram of how that happens. So you've got, and actually this is a very acidic environment, because high hydrogen ion concentration, which makes it very acidic. And so all of these hydrogens in the form of electrons and protons get trapped within this inner membrane space. Mitchell actually described how this happens, how these protons and electrons get pushed and trapped into the space, and they generate what he called a gradient, which means high on one side, low on the other, and the propensity is to try and reach equilibrium. And the ATP synthase helps with that. This molecule will then utilize the energy associated with with these protons and electrons, and the energy, the ATP synthase is then going to use the diphosphate to make a triphosphate. And it works like a rotor. It works like a big rotor. That's why they have it drawn like this. And that discovery of that protein defined how you can, start with a greasy donut and end up with adenosine triphosphate. It's a Nobel Prize winning experiment, actually. Okay, so no ATP synthase. It's a very important molecule for this entire process. It also is present in plants in their electron transport mechanism. Okay, so now you know cytochrome and ATP synthase. Okay, now let me go back to the and make sure that I've covered everything that I want to cover for this chapter. Now there was one other thing that's on here, and I'm going to go down to this figure right here. Now, I know that most people are going to just look at this and just totally ignore it. But this is just a summary of, how other kinds of foods, types of food are processed for energy. And we went through this, carbs. It's kind of like not very clear right now, but this is carbs, glucose to ATP synthase and energy. But this is proteins on this side, fats on this side. What I want you to know about this process is that everything gets broken down to what we call the monomeric units, like the individual monomers. The individual monomer, the individual unit for a carb, the most common individual unit for carbs is glucose. Okay? And that's what it's broken down to. And then it gets broken down even more during the cellular respiration. For proteins, they get broken down into individual amino acids. So proteins are made up of thousands of amino acids, and serine, glycine, isoleucine, valine, glutamine, all those amino acids. But then they go from amino acids and the amine group. You can't see this on it, but the NH2, the amine group, is separated, broken. So then it enters as an amino acid without the amine group. That's very important. Do you remember what I told you where that amine group goes? That is the molecule.
Oh, for the P?
For P, exactly. That NH2 is generated into urea, and urine gets its name from urea. Are you with me? So it gets broken down into that, well, not broken down, it gets incorporated into the structure it's used to make, for your body to make urea. And then urea is the major component in urine. It's called urine because there's urea in there and there's a bunch of other things in there. That's what makes it urine. It's got salts and some other stuff, and by combination, they call it urine. But the major compound in urine is urea. P. Okay? Are you with me? Okay. So that NH, and in class, I wanted to show you that, and I think I have a picture of, I thought I had a picture of, okay, so here it is right here. So here, this just shows this group. Now, all proteins have an amino group, and a carboxy group, okay? And this molecule, when you take away this molecule, Hello?
Claire, this is Kathy.
Oh, hi Kathy. How's it going?
It's going. We're out of the DNA buffer.
You're out of which buffer? The TAC?
DNA buffer.
Oh, the cDNA?
No, the DNA buffer to make the gel.
I figured you were gonna run out. Excuse me, ladies. I figured you were gonna run out, but I want you to make it yourself. Look on the shelf next to Emmanuel's bench. You see that? I want you to do it while I'm on the phone. Look on the shelf. Look on the shelf right to his bench. You're gonna see a bottle that says 50X. T.
Okay.
Are you there? Okay. Look in that shelf. It says 50X right there. You see it? Okay.
Got it.
That's it. So for a liter solution, how much do you need of that 50X and how much water?
For a liter?
Yeah, make a liter. I mean, just,
Oh.
yeah, 1,000 milliliters.
The 50X.
No, of 1X for the gel. Here's what I want you to do. I want you to take that 50X bottle. I want you to measure out 20 milliliters. 20 milliliters of that 50X, and then add the rest water, the water on the side.
Oh, okay.
Got it?
Yes.
Are you good?
Yes.
Okay, so you're gonna take 20 milliliters of the 50X. 20 times 50 is 1,000, so that'll be a 1X volume of a liter of buffer. Are you with me? Kathy?
Yes.
Okay, and then of course, use the clean water. Okay, are you good? Okay, bye. Okay, it's good I gave him my phone. I don't usually like to give students my phone number, sorry. But I knew something was gonna go wrong, I knew it. It was too much time with them alone. But that's how you learn, by doing stuff yourself. By doing it yourself. Okay, so this is that molecule that goes on to make urea.
Right.
Urea is the major molecule associated with urine. And I thought I put a picture in here of the structure of urea. And that way you can see how the amine group is the beginning of the structure. It's used to make it, but it's the major molecule in urine, actually. Okay, so, okay, and I want you to just know that all of these molecules are broken up. The fats are broken up to glycerol, because on the backbone of fats are made up of fatty acids, individual fatty acids, and glycerol. And they're just broken into, and then those get input into the cycle. Excuse me. Okay, and of course you get more energy from fats. Okay, now let's go to. Okay, so this is I think we're pretty much done with this chapter. So the next one is chapter 8. So let me pull up chapter 8. Let's see. Okay, now this is photosynthesis. I want you to know the equation for photosynthesis.